Hello, my name is Michelle Marr. I'm currently a PhD candidate working in Dr. Boyle's lab at the Colorado School of Mines in the Chemical and Biological Engineering Department. Today I'll be discussing my work in the analysis of the genome-scale metabolic model of Chromochloris zofingiensis. Our growing global population is driving the need to develop new, sustainable energy sources. To that end, microalgae have shown tremendous potential to be utilized as cell factories for the sustainable production of fuels and chemicals. They use sunlight and carbon dioxide to do this, producing a variety of value-added products. In these cell factories, we engineer pathways in a metabolic reaction network, similarly to how chemical production is designed with process flow diagrams for chemical engineers. This can get very complicated when considering a genome-wide scale, so we use computational models to aid in this process. Engineering these organisms for the overproduction of lipids or other metabolites of interest is difficult, and it typically requires a number of changes in gene expression across the genome. Metabolic networks are robust and tightly regulated to maintain homeostasis. So to perturb these systems to produce a product of interest, a broader understanding of the regulation and functioning of cellular metabolism is needed. Metabolic models provide a means of accomplishing this, allowing the metabolism to be studied in a more holistic manner. The figure to the right shows the design, build, test, learn cycle of metabolic engineering, an iterative process that is greatly expedited by the use of computational models. Creating metabolic models of algae is then the first logical step towards engineering photosynthetic cell factories. Stoichiometric models are advantageous in their simplicity as they can be implemented on a genome-wide scale, whereas kinetic models cannot. Flux balance analysis, or FBA, is a useful tool in stoichiometric modeling that has been successfully used in metabolic engineering of algae to assess fuel and chemical production potential and to predict gene editing strategies that would achieve a desired phenotype. In FBA, a system of mass balance equations is written for a given metabolic reaction network. This system of differential equations can then be written in matrix form. When the cellular metabolism is operating at steady state, as is the case during the exponential growth phase of a cell culture, this problem can be simplified into a linear algebra problem. A solution for the flux vector can then be calculated, providing a prediction of the flux through each reaction in the network, i.e. the reaction rate for every metabolic reaction in the cell. Chromochloris zofingiensis is a eukaryotic microalgae that has not only been identified as a top candidate for fuel and astaxanthin production, but it's also been shown to grow in a wide range of industrially relevant conditions, such as outdoors, using wastewater as substrate, urea as a nitrogen source, and in a mixed culture system. Astaxanthin is a carotenoid that has been suggested to serve as a photoprotection for the cell. This compound is a very high-value antioxidant, and it can be sold for an estimated $7,000 per kilogram. When grown in the presence of glucose, this organism performs a unique and interesting switch from autotrophy to purely heterotrophy. This switch is characterized by the breakdown of photosynthetic machinery and the degradation of chlorophyll, even while under continuous illumination. During the resulting heterotrophic growth, the cell stores energy in the form of lipids and starch. The figure to the left shows this difference, where the heterotrophic cultures of this organism are orange from astaxanthin content. The figure to the right shows this switch with illustrations reconstructed from cryosoft X-ray tomography data. The annotation engine RAST, standing for Rapid Annotation Using Subsystems Technology, has been used to facilitate the construction of genome-scale metabolic models in model systems like yeast and E. coli. But the system functions poorly when it's applied to photosynthetic systems. This necessitated the development of RAPS, standing for Rapid Annotation in Photosynthetic Systems, an automated annotation engine that can produce new genome-scale metabolic models of algae from genome data and donor models. This system was created by Alex Metcalf in our lab group. So here's a brief schematic of how RAPS works. A list of proteins that are predicted is compiled from genome sequence data. This list is then compared to a database of metabolism-associated enzymes from the donor models using the NCBI protein blast tool. If the E-score is satisfactorily low, indicating that both proteins share a function, then the associated reaction is added to the draft network reconstruction. Historically, the process of creating a genome-scale metabolic model is a tedious process done by hand. New models are published, adding reactions to older models, and through this iterative process, the model becomes more and more complete with a higher number of reactions included in the metabolic network. With the use of RAPs, our first draft genome-scale metabolic model of Chromochloris has a higher number of reactions than other algal models reviewed. So by leveraging new computational tools like RAPs, we can accomplish a lot more in a shorter period of time than was possible in the past. Here's an overview of the genome-scale metabolic model of Chromochloris zofingiensis that was created from RAPs from Chlamydomonas reinhardti and Nanochloropsis gaditana donor models. The output model from RAPs was modified slightly to include reactions for astaxanthin production, as neither donor models had this capacity. In this figure, panel A illustrates reactions shared with donor models, and panel B illustrates the localization of metabolites and reactions across 8 cellular compartments. Because this organism displays very different behavior depending on the carbon source used, 3 different biomass equations were constructed to describe autotrophic, mixotrophic, and heterotrophic growth. These biomass equations were built from detailed experimental data on the biomass composition in each of these 3 conditions. A variety of methods were used to quantify macromolecules. Each method was optimized for its application to these tough cells. The results of this analysis are shown in the figure here, where TP represents autotrophic growth, TAP represents mixotrophic growth on acetate, and TGP represents heterotrophic growth on glucose. Mixotrophic growth on acetate and autotrophic growth both have highly similar biomass compositions, as might be expected from observed similarities in their physical appearances and growth characteristics. The heterotrophic cells, however, show noticeably higher lipid and starch contents, while the protein and chlorophyll content is lower. This loss of photosynthetic machinery in heterotrophic C. zofingiensis cultures has been reported, and this might contribute to the lower protein contents observed here. This accumulation of energy storage compounds is also well documented in the literature. Results presented here, with lipid content at approximately 38%, fall within an expected range of 30 to 52% lipid content reported in the literature. Cellular lipids were saponified and methylated to create volatile fatty acid methyl ester derivatives, and these FAMEs were quantified via GCMS. The results of this analysis are presented here. One interesting observation I'd like to point out is the far higher content of oleic acid for heterotrophic cultures. This observation has also been reported in the literature. One study on heterotrophic C. zofingiensis cultures found that the introduction of glucose drastically increased the expression of 2 genes involved in oleic acid synthesis, and this might explain this relatively high oleic acid content observed. Cellular proteins were hydrolyzed and the resulting amino acid mixture was derivatized for analysis on the GCMS. These results are presented here. Even in terms of amino acid composition, there were significant differences in biomass composition between the different growth conditions. And these changes that occur simply as a result of changing carbon source highlight the need for experimental measurements of biomass composition in this type of study. All of this experimental data was used to create 3 different biomass equations for C. zofingiensis in autotrophic, mixotrophic, and heterotrophic growth. The genome-scale model of C. zofingiensis and these newly constructed biomass equations were used along with experimentally measured constraints to conduct a FBA for each growth condition under continuous light. These conditions consisted of growth rates as well as uptake rates of glucose and acetate for heterotrophic and mixotrophic cultures. Here are the results of this analysis, with the autotrophic growth in TP media on the left and mixotrophic growth in TAP media on the right. In these flux distribution maps, the thickness of the arrow indicates the rate of the reaction that said arrow depicts. Pictured is the predicted flux distribution through the central metabolism of C. zofingiensis. Both flux maps appear very similar, as expected from observations of their similarity in biomass composition, growth, and physiology. However, for the TGP cultures containing glucose, the results were very different, and here things started to get a little bit weird. According to model predictions, the measured glucose uptake rate far exceeds the amount that would be needed to match experimental growth rates. If all of this carbon was being used in respiration, the growth rate would be a full order of magnitude higher than was observed. So this excess carbon had to be going somewhere else, but where? Well, turning back to our computational model, when this model was forced to match both experimental data for glucose uptake and growth rates, our computational model predicted the formation of fermentation products. This was primarily in the form of lactate and ethanol. To further investigate this finding, spent media samples from mixotrophic TAP cultures and heterotrophic TGP cultures was filtered and analyzed via UPLC-MS. These results are presented here. An untargeted metabolomics approach was first used to analyze for any secreted products that might be in the media, so that our results are not limited by our own model predictions. As a result of this approach, absolute quantification of identified metabolites was not possible, so results presented here are with relative quantification. The size of the lactic acid peak, only present in the TGP cultures, indicates that lactic acid was indeed being secreted to the media. Additionally, a lactic acid enzymatic assay kit was used to then verify the presence of lactate in cultures grown on glucose. Through these two independent methods of analysis, lactic acid excretion was then confirmed. In conclusions, the variation in carbon source has a tremendous impact on biomass composition, especially with organisms like C. zofingiensis. And this has strong impacts on the flux distributions predicted. Our genome-scale metabolic model of C. zofingiensis was used to make predictions of growth in 3 different carbon sources. Our simulations predicted that excess glucose uptake is sent through fermentation pathways. This is a novel prediction that was then confirmed using experimental evidence. Future work will involve collecting a detailed experimental data on lactate excretion over time in glucose-containing cultures. Additionally, I will be performing an isotopically assisted metabolic flux analysis using 13Z labeled glucose tracers. In this experiment, isotopic labeling patterns of intracellular metabolites will be used to back-calculate fluxes through the metabolic reaction network, which will provide experimental backing to our computational estimations of cellular flux. So finally, I would like to acknowledge my wonderful lab group, our project collaborators, and our funding sources. Thank you so much for listening.